Hello, good evening, everyone. You're welcome back to the channel. Today we are discussing bacteriophages, and we're going to discuss part one of a two-part series on bacteriophages. What are bacteriophages? Bacteriophages are viruses that inhabit and like to eat up or take up space within viruses. Sometimes they are simply called phages or bacteriophages. So they are important, and they are numerous in nature, and they're important to know because they shape bacterial population, because they're always invading bacteria and using up the genetic machinery and genome of the bacteria to make many copies of themselves. They are well known in nature. They are also ubiquitous in nature, and they help to shape bacterial population. Now, the bacteriophages, remember I said they are viruses. So the question is, what does their structure look like? They have four basic elements within their structure, which is important for us to know. Because they are viruses, they have the capsid, they have the viral genome. Now they have the tail pins or tail fibers, and then they have the base plates. These are four structures that you have in the bacteriophage, and it's important to know. First, the capsid. This is the protein coat. It's made of protein or protomeres, and it is important. It surrounds or it covers the bacteriophage. Now, the capsid can have many different types of shapes. It can be a polyhedron, it can be an icosahedral shape, many different types of shapes that the capsid has. And that's the first thing, the capsid. The second one is the viral genome. Now, like I said before, the viral genome can be a DNA or an RNA, and it could be single-stranded DNA or double-stranded DNA, single-stranded RNA or double-stranded RNA. So that is the viral genome. So you have the capsid, you have the viral genome. Thirdly, you have the tail pins or tail fibers. Now, these are fibers that we can see all on the structures of the bacteriophage. And the importance of these structures is that they are able to help the bacteriophage attach themselves to receptor sites on the bacteria. Attach themselves to receptor sites on the bacteria, and this is important. Now, after the tail pins, you have the base plates. The base plates are also important because it is through the base plates that the bacteriophage are able to inject their viral material or viral genome into the bacteria, where they can now go there and start to make many copies of the viral genome within the bacteria that they have invaded. So this is important to know. Now, the life cycle of the bacteriophage is also similar to the life cycle of viruses, and we've discussed the life cycle of viruses before. So just something similar is what we have for the bacteriophages. The stages in the life cycle of the bacteriophages: you have attachment, you have penetration, attachment, penetration, then you have replication, and then you have assembly, maturation, and lysis. So I'll take them one by one. Attachment. Like I said before, the tail pins or tail fibers help the bacteriophage attach itself to receptor sites on the bacteria. So when this occurs, this is attachment. Now the next step is penetration. And in penetration, you have these base plates that are on the bacteriophages coming down, and through the base plates, you have the injection of the viral genome into the bacteria. This injection, this process of injecting the viral genome into the bacteria is what we call penetration. Now, after penetration, we have replication and transcription. Now, in replication and transcription, what we have occurring there is that this viral genome that has found its way into the bacteria begins to replicate, make many copies of itself while it is within the bacteria. And it makes these many copies using the genetic material of the bacteria as the raw materials with which it will make many copies of the viral genome which has been injected from the bacteriophage into the bacteria. Now, the next step is assembly. While now, when you have many copies of the viral genome which have been made within the bacteria, you now have capsid being assembled around all these various different viral genomes which have been replicated and have been made within the bacterial cells. So you have capsids forming around all these different viral genomes, and this is what we refer to as assembly. Now, when all of this is done, of course, when the bacteria, all of its genetic material or machinery has been used up, the bacteria cannot sustain life again. So the next thing that occurs is lysis. So there's a break in the cell wall and in the integrity of the bacteria, such that all these new bacteriophages which have been formed by capsid being formed around the new viral elements or viral particles that have been replicated from the original bacteriophage, they are all, when the bacteria lysis, all these new bacteriophages are let out and they are able to come into the environment. So this is the life cycle of the bacteriophage. You have attachment, you have penetration, you have transcription and replication, you have assembly, and then you have lysis. So thank you very much for listening to this video, and do have a wonderful day.